So I'm going to go ahead and get started and move on to our panel discussion and our panel looking at advancements in the treatment of PKU. What we're going to do is each panelist is going to come up separately because they want to see everybody else's slides out in the audience. And so they're going to come up separately and give their presentation, and then afterwards they'll all come up here for the question-answer period before we break again. And this panel is moderated by Ray Stevens, who I have the pleasure of introducing. And I feel just extremely fortunate, to be in this position with 2 children having PKU, and feel very blessed to be a part of the National PKU Alliance as the executive director. And one of my highlights this past year was actually having the opportunity to sit down with Ray, tour his lab at Scripps, and learn really firsthand about the work that his lab has done and the work that they are ongoing for treatment of PKU. For those of you who do not know, Ray is one of the leading researchers when it comes to PKU treatment. In 1997, he discovered the structure of phenylalanine hydroxylase, which is the protein responsible for PKU, that led to the development of Kuvan and also PEG-PAL. And so we are so fortunate to have him here with us. I've had the also pleasure of presenting with him at a few of the different state walks this past year, and we're just so pleased that he is a part of our community for the difference he's already made in the lives of my children, the lives of children here, and adults as well with PKU. So thank you, Ray.
Thank you, Christine. God, this is intimidating. Seriously. I've actually, over the past probably 10 years or 7 years, I've visited almost every state, going to different PKU walks and actually getting to meet a lot of the sort of people that are throughout this room, but actually seeing everybody together. And to put this in perspective, a National PKU Alliance or Association, it was actually tried 6 years ago, 5 years ago, and it was always difficult, to get everybody together. And I have to give, Dick Michaud and Christine and everybody tremendous credit. What they pulled off is absolutely amazing. So a huge thanks to Dick for doing this. Now, I also have to put this in perspective. We, I think, in terms of the PKU community, sometimes lose the perspective of basic science, drug discovery, what it takes. Many disorders, diseases, people will work on these things for long, long periods of time. 10 years ago, there was absolutely, as far as I know, no real therapeutic options that were really taking place. Dick Koch, who's getting one of the PKU Hero Awards today, is one of the pioneers together with Charles Schreiber. But 10 years ago, in the year 2000, there weren't a lot of options. A lot of basic science was going on. The maternal PKU program at the NIH that Dick was organizing was still going on, but there were really no options out there. And so this slide to me sort of summarizes, that there were no therapeutic options, but today, 10 years later, we actually have multiple therapeutic options now available. And the great thing is, and these are things to help quality of life. This includes some of the speakers that you're going to hear about today. Denise Ney, who will be talking about the improvement of the foods. Barbara Burton will be talking about Kuvan and PEG-PAL and the monitoring devices and stuff. You hear Ira Fox will talk about the future and cures that can actually take place, that can actually take this one level even further. And Catherine Mosley will also be talking about alternative options with large neutral amino acids. But none of this in the past 10 years, absolutely none of this could have happened without the combination of PKU researchers, families, industry such as BioMarin. All these different pieces, the dietitians, industry, researchers, families, the fundraising, MacPad, who was one of the sort of pioneers in raising money. The Michaud family and the Tuxes for Tia, that previous slide that I showed was a picture of Tia Pizziali in that very first Tuxes for Tia event 5 years ago, Allison reminded me. All those things had to come together in order to really advance things. And this is where I think the theme, high hopes, That was the momentum. That got things going. What they've accomplished over the past few years really got things going, and now I think it's going to go to an even better level because of events like this and because all the other issues that are now starting to be addressed. So this is really a story, a huge success story in basic science and in drug discovery about addressing something that needs to be addressed, PKU. Now, this right here is a movie where what you're looking at, as we all know, and I'll talk a little bit about the sort of basic science behind this, this is the protein phenylalanine hydroxylase, where you get a mutation in this protein, and that then leads to phenylketonuria, PKU. This right here is the liver. This is where you're supposed to be degrading, so for the kids in the audience who like the movies, I like movies too. I'm going to see Avatar tomorrow night. And so this enzyme, BH4, that we know as Kuvan, stabilizes the protein. It's a requirement for the enzyme itself to work. It then brings in phenylalanine. It's able to then, ordinarily, if this is working, you have phenylalanine that hits. It then gets converted into tyrosine. This is the way that it's supposed to work. And then when you have mutations in this large part right here, that's where, in fact, you're not able to convert phenylalanine into tyrosine. And that's why we needed to have alternatives. Now, this picture, for most of you, this is going to look like spaghetti, colored spaghetti. And this is actually what I do for a living. I try to create and understand the structure of the protein phenylalanine hydroxylase. And so it's composed of what we call the regulatory domain, speeds up and slows down the enzyme, the catalytic domain that causes the phenylalanine to be converted to tyrosine, and then the tetramerization domain, and I'll show you a picture of that in a moment. And so we study, this is what we determine the structure of this in order to understand how this protein works, and then when mutations in this protein affect, what exactly the causes are. This is the tetramer formation from this, and so the reason that I actually show you this picture is really to highlight and to help you understand how does Kuvan work. This spaghetti-like structure is incredibly unstable. It is like a piece of spaghetti. It unravels very, very easily in our bodies if you make a mutation. By putting Kuvan in there, or BH4 tetrahydrobiopterin, I like BH4 because it's easier to say, or Kuvan, what it does is it binds there and it actually stabilizes it. It's like a pillar of a house or a beam. It stabilizes that and allows the protein to work for a longer period of time. Now, that right there was particularly sort of Kuvan. I'll actually skip this slide here. And when we first solved the structure, and I'm often asked the question, I'm going to take tangents because I'm a scientific geek, and so I am allowed that sort of luxury. When we first solved the structure of phenylalanine hydroxylase, we were interested in the basic science behind it. And I actually got to confess, I did not know very much about PKU. We published this in a scientific journal, and I think it was 1996, 1997, and I got this phone call from Dick Koch. And Dick said, you know, we're having this meeting at a hotel in Los Angeles Airport, the maternal PKU advisory meeting. And he asked if I would come down and explain the results. I sat around the table with dietitians, the nurses, some of the sort of family representatives, the clinicians. And to me, if anybody asked me, why did I get into PKU research? It's because of one of our PKU heroes tonight, Dick Koch. You know, his inspiration, watching him. His inspiration in watching what he did over decades of time towards PKU was absolutely inspiring to me. And that made the connection. And from that point, I decided, besides doing basic science, I want to actually see if I can make a difference and try to help people towards that have PKU. So one of the first things that we did that actually, you know, Dick, you're the one that prompted us to look at this, is we wanted to know from this protein that I showed you in that movie, when you have mutations in this protein, what happens? We thought all the mutations would be just in the little factory part where phenylalanine gets converted to tyrosine. In fact, the mutations are all over the place. You mutate this protein almost anywhere, it falls apart. Some mutations, mild PKU, have gentler effects. It allows it to be, you know, it'll actually stay as a protein. Some of the mutations completely destroy the protein itself, classical PKU. So now, because of the basic science behind this, we understand this. Again, only in the past 10 years do we now understand the structure, what happens when mutations affect the protein itself, and we now understand exactly how this protein, this enzyme, actually functions. So it's been a tremendous breakthrough at the basic science level. Now, what about the sort of non-dietary PKU treatment options? One of the first things that we thought after, Dick inspired me to sort of start thinking about this was, naturally, as a chemist, I thought, let's make a small molecule, something that was a big protein, so something that's more like a regular drug, aspirin, or some other drug that you may know about. And we thought, let's design a small molecule that may activate this or inhibit it. However, we couldn't come up with any ideas. We were stuck. And we actually tried really hard with a lot of smart people, but we really couldn't come up with any ideas. And then the idea started emerging, and I'll highlight this in the coming slide, about stabilizing molecules like Kuvan, BH4. We also thought, what about the injection of an enzyme replacement treatment, so an injectable therapeutic of the actual protein itself? This was prompted from 2 points of view. Our other PKU hero tonight, Charles Schreiber, came up with the idea in 1998 about injecting a protein or orally available protein for use. I have to confess, separate from that paper, I was in the grocery store and I saw this stuff called Beano. I don't know if you guys know what Beano is for, but I had really bad gas. I probably shouldn't say that, but I had bad gas. Okay, fine. And I was just sort of looking at it, and as a chemist, sort of scientist, I'm like, all right, what's in this stuff? And it turns out it's an enzyme. You're actually taking, Beano is beta-galactosidase, I think. Something like that, I can't remember. But it's an enzyme, and I thought, well, if you can do that with an enzyme and Beano can take care of my gas, then maybe there's a connection. Follow me, there is a connection there. I knew I shouldn't have gone down that path. Anyway, and then we thought, besides the injectable, what about an oral enzyme replacement therapy? The real, to me, the real sort of possibilities. So now taking a step back, stabilizing mutations. The real heroes for this are people like Professor Cure in Japan, Charles Schreiber, Dick Koch, Ninan Blau, Ruben Madelon. These are all guys at the very early stages, took a chance on this, and these were experiments to try and see whether or not a molecule like BH4 or Kuvan could actually help to lower Phe levels in people. They were pioneers. They tried things because they saw the need. Now, and what our contribution has been to follow what they've been doing and to try to understand exactly how these mutations take place. So again, this is the picture of the enzyme, the catalytic domain, the regulatory, and the tetramerization. The way that these things work, so many people are always asking, how does Kuvan exactly work? As I said, this protein is quite unstable. It falls apart. It's like spaghetti. Kuvan is like, as I said, a pillar or a beam in a house. So when you have this right here is the Kuvan itself, if you make a mutation, BH4 can no longer bind into that site, or it's destabilized, or it has a lower activity. And so by having higher concentrations of Kuvan or BH4, it can therefore make this enzyme work. Make it work as a factory, so phenylalanine will start getting converted into tyrosine. So there's 2 different types of mutations. We published this work actually together with Dick and Charles and many other researchers in the field of PKU, and determined that about 20% of the time, Kuvan is working to help increase the activity of the enzyme, and about 80% of the time, it's actually helping with the folding and the stability of the enzyme, keeping it in the body longer so it can actually work. Another question that we've been asking, and I've talked to lots of people over the past few years at a lot of the PKU walks, and encouraging them to get genotyped. So what does genotype mean? It's actually very important for basic science reasons for us to understand when people do respond to Kuvan, what mutations do they have in their bodies? One of the most popular mutations, things like the tryptophan. Let's see if I can sort of point this. There's a tyrosine 414 cysteine mutation, quite responsive. There's some other mutations that are less responsive. We need this information so we can actually someday be able to correlate where will Kuvan help people? Some people are not responsive to Kuvan. Some people are responsive to Kuvan. Would like to be able to predict that in advance. So is there a genotype or what mutations you have and phenotype, the actual effect of a drug of Kuvan, what it'll have. So we're continuing to encourage people. In fact, we have a paper together with Charles Schreiber trying to understand this correlation, and it's still a work in progress. So we need more data, more information along these lines. What about those that do not respond to Kuvan? Now, Kuvan, you may not know this, and again, 10 years ago, it was just barely on the horizon. It was originally thought it was going to affect such a small percentage of people that it wasn't worth developing as a drug. I think the initial numbers were 10%, maybe 4 or 5%. But then now it's turning out it's a much higher percent. I think the numbers sort of vary. But it's, you have a very good shot, and everybody should try the BH4 loading test in order to find out whether Kuvan will help them. But we want to know for those people that do not respond to Kuvan, a percentage do not, what are the options for those individuals with PKU? And again, this is to improve quality of life, not the cures like we'll hear from Ira Fox and the newest fellowship applicant that's just been funded by NPKUA, but other ideas. So enzyme replacement therapy is being pursued to try to treat this. And this is an area where, we understood the system, we're starting to understand the disorder itself and the enzyme, but we really didn't have the funding to try some key initial experiments. And that's when Dick Michaud walked into my office at Scripps together with Ginny and said, our granddaughter Tia has just been born, and she has PKU, and I do a literature search, and your name seems to pop up in the PKU area. What are the sort of ideas? Can we actually help you do something together? And we're like, well, we're understanding this, would like to pursue this idea, but with funding in the United States, and most scientific funding is from the National Institutes of Health, where part of your taxpayer dollars go to. And in order to get funding within a place like the NIH, you have to basically have proof of principle. They don't tend to fund things high risk, unfortunately. You have to have enough data to convince them. And so Dick said, we're going to try to help you out here. We're going to try to raise the money so you can do these experiments and try things. This right here was our idea. This is the protein itself, phenylalanine hydroxylase. And we did some amazing science. We're able to take this protein and we're able to reduce it through protein engineering. We go in and we're able to change the atoms around, truncate this protein. We're able to make a protein that's this big to making it this small and really compact and super stable. And we were really proud of this. And we failed miserably. And this was actually tough on us. I didn't actually share this story with Dick. Now that we actually have PegPal, by the way, the first idea, Dick, didn't work. Just as a reporting piece. I'm sorry. But there's a reason to actually sort of be positive on this. And from this, we then went into the literature, and we had actually done this work together with Christine Sarkinson in Charles Shriver's laboratory. We then went in, and many of you know about genomic research. We've now sequenced the human genome. Charles Shriver tried this idea in 1998. It took us a little while longer. But we then went into the literature, everything that's been sequenced in the human genome, all the other plant genomes, all these freakish deep sea vent things that have been sequenced. And we asked the question, what are all the possible proteins in the universe that may degrade phenylalanine? Phenylalanine hydroxylase, the reason, as a therapeutic, we actually, I wouldn't necessarily say failed, but it has so many cofactors. It has that oxygen. It has that BH4. It has to have iron and everything else. As a drug, it would be very, very difficult to get to the FDA. So we said, what are the different molecules or enzymes in the body that degrade phenylalanine? And we looked at all the possibilities. And so this is genome research. And we came across this one, phenylalanine ammonilyase, and we were looking for things that would convert phenylalanine into something that wouldn't hurt your body, that would be non-toxic, or also that didn't have all these special requirements with it. And so phenylalanine ammonilyase would actually take phenylalanine, convert it into tranexamic acid and ammonia, and you just excrete it out. Not toxic at all. And so this is where the connection coming back to Charles, everything fit together really nicely and said, okay, maybe this might actually work. So the differences between this, phenylalanine hydroxylase, and this is an education for those of you that are in the PegPal trial, and we'll hear more about this from Barbara in the talk following, is that phenylalanine hydroxylase is from human. You're putting a human protein into a human. Straightforward. It catalyzes the right reaction, creates tyrosine. The disadvantages, all these requirements, and it's also a relatively unstable protein. That's why it leads to PKU. Phenylalanine ammonilyase does not come from human. It actually comes either from bacteria or yeast. So we do have an issue with the immune system wanting to, if you put this into a person, the immune system is going to want to get rid of it. We also want to make sure about what it's generating, these tranexamic acid and ammonia. And it does not produce tyrosine. So tyrosine supplements might be actually necessary for this. We were able to, I showed you the picture of phenylalanine hydroxylase. What my lab does is solve these very sort of, to me, beautiful pictures of these molecules. We can see all the different atoms and everything and where they fit. We solved this, and working very closely together with BioMarin Pharmaceuticals and, again, with Charles Shriver's laboratory at McGill, we came up with an idea to try to trick our bodies into not wanting to get rid of this protein. So we covered it with what we call polyethylene glycol, essentially a plastic that would wrap around the protein. And that would cover the protein itself so your immune system wouldn't look at it and say it's foreign and want to get rid of it or degrade it quickly. the search for this optimized molecule, this was an insanely intense collaboration, again, between my lab, Charles's lab, BioMarin, where we did a huge amount of work. I mean, if you think about this, we built, and I'll use a house analogy, 825 houses trying to find the right sort of system that would actually work. We tested 75 different molecules in an animal model with a PKU. There's a PKU mouse that has PKU that we do our initial experiments on. We tried multiple of these polyethylene glycol molecules, and we tried different types of administration routes to try to get this done. And again, from the initial work, from Texas for Tea event, and from MacPad, and from many of the other fundraisers that took place, we were able to actually collect enough data to then go to the NIH, the National Institutes of Health, and apply for funding. And then we were able to get a $1.2 million grant in order to take this even further, to do the right studies so that it could then go into clinical trials. And one could ask the question, you know, in terms of BioMarin's contribution, they did make a contribution. They were part of this. But as a business, it's very difficult in terms of their risk. And so we had to push this to a point where we really believed it was likely to work, not that it was high risk. So this right here shows the antibody response. By adding this polyethylene glycol, what you see is if you inject the protein itself, you generate a lot of antibodies. We cover it with this plastic completely. The immune system doesn't even see it anymore. These are the PKU mice, sort of different colors, and so we can actually see from their colors. Hope there's no sort of animal rights, but these PKU mice are absolutely critical for this type of research. They really, really help out. And yes, they are cute, and they're okay. What we actually were able to do, if you injected just a regular diet, no what was called PEG-PAL, so the PAL is phenylalanine ammonia lyase, the PEG is that plastic that surrounds it, so now that's the explanation for what PEG-PAL is. Nothing at all. You see Phe levels stay high. When we started adding Phe, this was over 120 days we did this experiment. You can see the consistent drop in phenylalanine levels for this. Again, more for classical PKU patients. And then we also did lots of experiments in, for example, both the brain as well as the plasma, trying to understand the phenylalanine levels. And again, we saw this very nice drop in this area. So, you know, that's what we've been focused on for the research. I actually consider this partly the past. You know, we have therapeutic options, things that will improve the quality of life. Kuvan, PEG-PAL, and again, Barbara will talk about this more, and particularly with clinical trials. But I want to wrap up this sort of overview with one more point, and that is, you know, there's now 76 years of PKU scientific research and progress that's been made, dating all the way back to Folling's description of PKU. Things like the Guthrie test, that's been absolutely critical for the field in terms of now understanding the patients that have PKU so it doesn't have to progress so long. Dick Koch and the maternal PKU studies that took place over multiple years really highlighting the importance and need of maintaining phenylalanine levels. And then we've seen a tremendous number of publications. In fact, there's now more than 5,000 publications around just PKU and phenylketonuria that are now in the scientific literature that have helped us to understand PKU and also the advancements of the technologies, the clinicians and their reports, and so forth. So it's been a tremendous, I'd say, journey and research that's gone into this area. So transitioning from that now, as I said, I consider a lot of that to be the past. You know, we're starting to see the clinical trials are coming out, the home monitoring devices that we're going to hear more about from Barbara Burton. Alternatives, large neutral amino acids. There are other options that are being pursued, and it's fantastic work that's taking place. Catherine Mosley will talk about the large neutral amino acid programs. The foods made with glycomacropeptides, Denise Ney. It's an absolute honor to hear that the NPKU, in terms of funding for the research coming up for this next period, it's a great portfolio that's been determined from the committee. You have future visions, transplantation, you have understanding the systems, Harvey Levy, and you have the food programs as well. And so Denise will update us on the foods made with glycomacropeptides, and then finishing off, Ira Fox will talk about the transplantation of isolated liver cells. So the last slide that I'm going to show is actually a special thanks from me personally, and that is I started off this talk describing how there's no one individual. I, by myself, as a researcher, I could have only pursued things so far. I could have studied the protein, and that probably would have stayed in the scientific literature and been used that way. Because of connections to people like Dick Koch, because of connections to people like Dick Michaud, Charles Shriver, and so many other PKU researchers, it's flourished. All of the donations, MacPad, all the other institutions and efforts that have taken place towards raising money for this type of research, and also the industry connection between BioMarin and NIH. None of this could have taken place without all of these pieces coming together. And so this is a success story, not of any one individual, but the whole collective, which is what NPKUA is all about. Thank you very much.